Gram staining is a crucial technique in microbiology that is used to differentiate bacteria into two major groups, that is gram positive and gram negative. This differentiation is based on the structure of the bacterial cell wall. Gram staining is a simple and a rapid procedure that can provide a preliminary identification of bacteria within few minutes. Gram staining helps to classify bacteria based on their cell wall structure, which is a fundamental characteristic used in bacterial taxonomy. The gram stain can provide insights into the physiology of bacteria. By examining the sample of the infected tissue or the fluid under the microscope, after gram staining, one can identify the type of bacteria causing the infection and select the appropriate antibiotics. Gram staining is an essential tool in research laboratories where it is used to study the structure, physiology, and behavior of the bacteria. Now, materials are required for gram staining. The bacterial cultures Staphylococcus aureus and Escherichia coli, microscopic slides, inoculation loop, crystal violet, that is the primary stain, iodine solution, the mordant, decolorizer, that is 95% ethanol, safranin, the counterstain, immersion oil, microscope. The principle of gram staining. The primary stain, that is the crystal violet, penetrates the cell walls of both gram positive and gram negative bacteria. However, the subsequent mordant iodine forms a complex with the crystal violet. The cell wall of the gram positive bacteria is composed of a thick peptidoglycan layer. The crystal violet iodine complex forms a large insoluble precipitate within the peptidoglycan layer, which is difficult to remove during the decolorization step. The gram positive bacteria appear purple or blue under the microscope. The cell wall of the gram negative bacteria has a thinner peptidoglycan layer surrounded by an outer membrane. The decolorizer, that is the alcohol, can more easily penetrate the thinner peptidoglycan layer and dissolve the crystal violet iodine complex. Since the crystal violet iodine complex is removed, the gram negative bacteria are counterstained with safranin, which gives them a pink or red color. Different steps involved in gram staining technique are heat fixation, crystal violet staining, iodine treatment, decolorization, safranin counterstain, microscopic examination, and interpretation of results. Now the step 1, heat fixation. Clean the microscopic slide with alcohol. Place a thin smear of the bacterial samples on the slide using a sterilized inoculation loop. Allow the cultures to air dry. Heat fix the bacteria by passing the slide over the Bunsen burner flame several times. Heat fixation prevents the bacteria from being washed away during the staining process. The treatment with the crystal violet. Flood the slide with the crystal violet for 1 minute. Rinse gently with distilled water. Then step 3, treatment with iodine mordant. Flood the slide with iodine solution for 1 minute. Rinse gently with distilled water. The iodine forms a complex with a crystal violet, which is retained by the gram positive bacteria but not by the gram negative bacteria. The step 4, that is the decolorization with ethanol. Flood the slide with a decolorizer, that is the 95% ethanol, for 30 seconds. Rinse immediately with distilled water. The control the decolorization time to avoid the over decolorization which can lead to false negative results. The step 5 is the treatment with the Safranin stain, that is the counter stain. Flood the slide with the Safranin for 1 minute and rinse gently with distilled water. Safranin stains the decolorized gram-negative bacteria pink or red. Gram-positive bacteria remain purple due to the retained crystal violet iodine complex. Now the microscopic examination and interpretation of the results. Allow the slide to air dry. Place a drop of immersion oil on the slide. Use 100x oil immersion objective. The appearance of purple or blue due to the retention of the crystal violet iodine complex indicates the presence of gram-positive bacteria. The appearance of pink or red due to the decolorization of the crystal violet iodine complex and subsequent staining with the safranin indicates the presence of gram-negative bacteria.